Evolution or intelligent design. What we're really talking about here in this section is the origin of what I call complex adaptations. Things that allow organisms to adapt to their environment that have numerous parts in place to accomplish that. And the question then becomes, how can all those different aspects evolve essentially independently, but in some sort of coordinated way? So several evolutionary principles are, she didn't give you one?
Let's take this.
So, nature selects from what they call random variations. Now, these are mutations. And nature doesn't actively select anything, so the term is a bit misleading. Those who are able to survive and reproduce at a greater rate are those that nature selects. That's all it is. And this is what's called mutation, natural selection. Now, in Darwin's time, he knew nothing about DNA, he knew nothing about genes. He just talked about variations. He had no idea how those variations arose, where they came from. So it was in the 20s, 30s, and 40s of the last century where various evolutionists were able to piece together that it's DNA mutations that provides the stuff for evolution to act on. Then, I already mentioned this, an organism strives to survive and reproduce. Surviving is not enough. And supposedly those who have a slightly better adaptation to that current environment will survive better and hopefully reproduce more prolifically. And so that grouping, that genetic structure will become to dominate the population over a period of time. It's important to realize that it's the species that persist through time, not individuals. There's already been plenty of discussion, and we'll talk about it at a later presentation in this class, but there are those who want to do human genetic engineering. In other words, be able to change the genetic structure of a human being and be able to pass that on to offspring. There's already been a scientist in China who's done exactly that. There are a few genes that are known to offer resistance to the HIV virus, and he cloned those genes into a pair of embryos, allowed them to be implanted, and now those children have been born. Now, he's been severely punished. He lost his job. He was put in prison for a period of time, but he's back working in the lab again, so who knows what he'll do next. There have been many attempts to try to clone a human being. So far, all those attempts have not succeeded. We've cloned cattle and sheep and pigs and dogs and cats and all horses and all sorts of things, but for some reason the human being proved rather difficult. We do know that in the late 90s, early 2000s, there were several who were making the attempt. They were trying to clone a human. But it's not the individuals that count, it's the species. And if somehow it's determined that certain individuals of a species, including human beings, are not going to represent the species very well, then their value decreases. And we even had laws in place, and I think about 31 states here in the U.S. that started around the 1920s, 1930s, that sterilized involuntarily individuals the states thought should not reproduce. Without their consent, they were sterilized. Nazi Germany put together some of their earliest laws based on the laws the U.S. had. Some of these didn't come off the books until the 1960s. So, that's even been tried here in the U.S. to attempt to regulate the genetic structure of the species. And individuals at that point don't count for so much. So, you know woodpeckers, right? Can anybody identify these? I didn't think so. The one on your left, the black and white with the red top of the skull, is a pileated woodpecker in the U.S. The woodpecker on the right is called the green woodpecker, and it's from northern Europe. Now, woodpeckers are strange creatures. They bang their heads in the trees. Anybody know why they do that? Why do they peck at the tree?
Isn't it to get bugs out of the tree to eat?
Get bugs out of the tree. Sometimes they do it to create a nesting hole, but more often than not, they're simply hunting. Now, in order to do that, the beak has to be quite hard. Can't be fragile or brittle. It's got to hold together. So it is. Notice with both woodpeckers, notice at the bottom, they have their tail feathers pushed up against the tree. Now they have, their toe structure is a little different. Most birds have 3 toes in the front, 1 toe in the back to grasp a perch. Woodpeckers have 2 toes in front, 2 toes in the back to grasp the vertical surface of a tree. And then, so they have 2 feet that are able to hold onto the tree, and then they use their tail feathers as the 3rd leg of a tripod. And now what we're going to talk about mainly is the tongue of the woodpecker, but I want to mention just a couple other things here. That the whole skull and the back of the beak and front of the skull is all engineered, I use that term advisedly, in such a way that most of the shock of hitting the tree goes down the spine and back into the tree through the tail feathers. So it simply redirects the shock. It doesn't get headaches. It doesn't get concussions. At the rate and the force with which they hit the tree, if that was a human skull, we'd all be dead. We can't survive the kind of impacts that the woodpecker is able to. Now, about the tongue, also when they're hitting the wood and there's splinters flying away, they have a small, what's called nictitating membrane that automatically covers the eye, so they don't get any pieces of wood in their eye. It happens automatically. They don't cause it to happen. So, some woodpeckers are able to extend their tongue 3 to 5 times the length of their beak. Sometimes it looks kind of like that. Now you have to ask the question, okay, when it comes back in, where does it go? What do you do with it? Well, it looks like this. It's from a museum. And you see on this side, there's the tip of the tongue. And it comes back, you see it splits in half. And one half goes around behind the skull this way, and the other half goes around the skull and comes over this way. And it comes back together here at the right nostril. And this extends into the upper beak. So when the woodpecker wants to stick out its tongue, well, first of all, this attachment loosens and it attaches between the eyes. These big loops flatten down and out goes the tongue 3 to 5 times the length of the beak. Now, I have a whole 20-minute presentation on woodpeckers, but that's for later in the year. And so what I'm going to show you here is this comes from a ornithology study of birds textbook. I got permission to put it in my book, Natural Limits to Biological Change. And so you can see here is the tip of the tongue. So we're going to go backwards from the tip of the tongue. Goes into the throat, makes a big loop in the throat. Goes up around the back of the skull. Then comes, next picture, comes down between the eyes and inserts into the right nostril. Not the left, the right nostril. Don't know why that makes any difference, but that's what happens. And so when it wants to stick out its tongue, again, as I showed you, this connection loosens and reattaches here between the eyes. The big loops flatten out, and out goes the tongue. So you have a question here. How do you evolve a tongue like this? No other birds bang their heads in the trees. Only woodpeckers do that. So something happened to give them that ability. But the tongue, they have a long tongue so that they can go in find little tunnels that insects and larvae are in and stick their tongue up and down the tunnel, and they have barbs on the end of their tongue and a sticky saliva, and so everything sticks to their tongue. And so that's how they get their food. But how do you evolve something like that? Because you have to do two things at the same time. You have to elongate the tongue, and at the same time you have to devise a retraction mechanism. Because if you elongate the tongue without a retraction mechanism, it could lengthen out beyond the tip of the bill, and if you try to drill a hole, you cut your tongue off. That's not very helpful. So you have to do two things at once. Evolution is never very good at doing two things at once. So, I just talked you through that. Mrs. Bohlin added this. These evolved two completely different systems at the same time, a longer tongue and a retraction system. Again, very difficult for evolution to do both those things at the same time. More questions of evolution. What do you think those little bugs are? See them? Here?
Dung beetles?
Dung beetles. They look like dung. Therefore, birds won't try to eat it because they think they're nasty. It's just little small pieces of turd. The question is, how can you evolve that? Because at the earliest stages, they're not going to look anything like a piece of dung. So as Stephen Jay Gould, a former evolutionist from Harvard University, said, can there be any edge in looking only 5% like a turd? No. There offers no protection. So there's nothing for nature to select. There's no advantage yet. You have to probably get at least beyond 50, 60, 70% before there would be any benefit. And then you might be able to perfect that further, but getting started, slow, gradual, just doesn't work. Now, you've all had biology, correct? Do you ever study the peppered moth? No? No peppered moth? If you took biology from me, we talked about peppered moth, but...
We did?
Yeah, we did.
I might have forgotten that.
Yeah, I figured you would. The peppered moth is a species of moth in England, and there have always been two varieties. You see in the upper left, there's a moth there that's very speckled, and it's very well camouflaged on the light tree bark of a birch tree, which has both light and dark patches. The other form was called a melanic form, and it's just completely black, and it is not the least bit camouflaged. Now, throughout insect collections throughout the years in England, always far more peppered variety than there were of the melanic form, but the melanic form was always there. Then with the Industrial Revolution, the beginning of burning of coal, sometimes these birch tree bark would get covered with burnt coal dust, or sometimes the lichens on the trees would just die. And so suddenly on the bottom figure you see that suddenly the peppered variety was more conspicuous and the melanic form was hidden. Now it does demonstrate natural selection. It seems to, at least, because as things changed, the frequency changed. This study was done in the 1950s, and it was the first time that they'd ever studied something called natural selection in the wild. So that's why it was a big deal. You can find it in every high school, college textbook. They'll always talk about it. All it does is document a change in frequency. but there's no new adaptation. Both forms still exist. Both forms have always existed. Simply now, the melanic form outnumbers the peppered form. Now, also, they discovered later that these moths never alight on tree trunks. They alight on the undersides of leaves, which are green. Neither has any camouflage effect. These moths you see here were glued to the tree. Those aren't natural photographs of any kind. So they're not really sure if there actually was natural selection occurring. Why the frequencies started to shift now, they're just not really certain at all. So one more good example of natural selection kind of goes by the wayside. Here's a poor Drosophila fruit fly. This is showing you various locations of mutations on chromosome 2 of fruit flies. Some can have bent wings, some can have a brown eye, some can have a very small vestigial wing. Sometimes they can have purple eyes or light eyes or dumpy wings or star-eyed. We know all those mutations. We know where on the chromosome they're located. We've done lots of nasty things to fruit flies over the years. We've exposed them to X-rays, to chemical mutagens, all kinds of things to cause mutations. And we've made some pretty strange looking fruit flies. But in the end, they're all still fruit flies. We didn't change fruit flies into anything else other than basically damaged fruit flies that could not survive in the wild. They could survive in a laboratory, but not anywhere else. So Pierre-Paul Grasset was a French evolutionist, and he wrote a book that I quoted in my book, Natural Limits. He was talking about the mutations of bacteria. He says, what's the use of their unceasing mutations? A swing to the right, a swing to the left, but no final evolutionary effect. Mutations don't do anything. And that was even in the early 20th century. Now, this is a painting by Karen Quinto, and she wants to demonstrate what's called the Cambrian explosion, which we're going to talk about. And what she tries to show you is that over on this side is the beginning. You have very few forms available, but then suddenly as you go through the time lapse of the Cambrian period, all of a sudden all these different forms, in this case all these different colors, just suddenly appear. Now, what would have been better, however, more accurate, is if you did it this way. Because in the Precambrian, all you have is this, almost nothing. And suddenly in the Cambrian, all these new forms suddenly appear. There is no gradual development of these things. My wife figured out how to do that. I would never been able to figure that out. So, Cambrian explosion. Here you see some of the creatures of the Cambrian. The Cambrian is located here at the very bottom of the geological column. And you see right below it is what's called the Precambrian. And you see the Cambrian supposedly began, you see here it says 570, that means millions of years ago. And then it lasted till about 492, so about supposedly 80 million years is how long the actual Cambrian period lasted. Now this is what evolution's story of life is supposed to look like. You start off with one life form that somehow came together through chemical reactions in the early Earth, and then slowly it would branch off into maybe different species, but they're all still quite similar. And then those species would evolve some and they'd form what we call genera. And then they'd evolve and form orders and then classes, and then finally phyla. So you don't get to the phyla until you get way at the top. But everything had to be pretty similar at the beginning. It's now what we found. Time Magazine, 95. New discoveries show that life as we know it began an amazing biological frenzy that changed the planet almost overnight. Now, almost overnight means geologically overnight. And we're talking about just a few million years here. We'll talk about that in a little bit. Time Magazine again, virtually everyone agrees that the Cambrian started almost exactly 543 million years ago, meaning the Cambrian explosion. And even more startling that all but one of the phyla in the fossil record appeared within the first 5 to 10 million years. Geologically speaking, using dating methods, you can't navigate anything smaller than that. You can navigate maybe 5, definitely 10 million years, but anything within that, you can't. I mean, it could actually have been overnight. It could have been. Because you can't discern things that small. Simon Conway Morris is a Christian geologist, studies the Cambrian. So the term explosion should not be taken too literally, but in terms of evolution, it is still very dramatic. What it means, now listen carefully here, what it means is rapid diversification of animal life. Rapid, in this case, means a few million years, rather than the tens or even hundreds of millions of years that are more typical. So clearly things are happening very quickly. When I showed you that life story for evolution, the branching and all that, I told you at the beginning things would just go from species to genera to orders and develop the higher classifications later. Well, here the highest classification appears first. The phylum appears first. Even more speculative, Time Magazine again, are scientists' attempts to address the flip side of the Cambrian mystery. Why this evolutionary burst, so stunning in speed and scope, has never been equaled. Why do no new phyla show up in the rest of the fossil record? Or just talking about animal life. There are some species that are so small, we have no fossils of them. So you can't tell when they came into existence. But now most paleontologists believe that even all of those small animal phyla also appeared in the Cambrian. So we're talking about phyla here. How different are they? Well, what you see here, you see a dragonfly, a crab, a bee, and a lobster. And the phylum is called Arthropoda. Insects, crustaceans, centipedes, etc. They have their skeleton on the outside. They all have an exoskeleton. They all have discernible body segments. And some have wings, some don't. But they're easily recognized as arthropods, jointed appendages. Arthro, Latin root, any idea? Poda means foot.
Well, arthritis, I think it means joint.
Joints.
Yeah, arthro, I
Okay.
believe is jointedness.
So, jointed appendages. A different phylum is called Chordata, and that's the phylum we are in: fish, amphibians, reptiles, birds, and mammals. Well, our skeletons aren't on the inside, they're not on the outside, they're on the inside. We have an endoskeleton, not an exoskeleton. Now, supposedly, these two phyla are evolutionary connected, but both show up in the Cambrian at the same time with no intermediate suggested between them. All the different phyla that show up in the Cambrian have not only no ancestors in the Precambrian, but there's nothing indicating who evolved from what. Understand phyla? Okay. Samuel Browning, geophysicist, also from Time Magazine here. We now know how fast is, and what I like to ask my biologist friends is, how fast can evolution get before they start feeling uncomfortable? What he means here is that at some point, this is just too fast for this large amount of change to occur across the board of the animal kingdom. And it just, we know it was fast, and he's a geophysicist, so he's not a biologist. So how fast can it get before you start feeling uncomfortable? Trust me, they're already uncomfortable. So how significant was the change? Well, these are little tufts of cyanobacteria, and they lay down some limestone-like deposits, and these are called stromatolites.
How big are the?
Oh, they can be about this big around. Okay.
So little Miss Muffet sat on a tuffet would be like that.
Yeah.
Okay.
Maybe this one down here, but you know.
Okay.
This is the only place in the world now that we find stromatolites, but they were very common in the early Earth. And they look exactly the same. They haven't changed, supposedly, over billions of years. For billions of years, simple creatures like plankton, bacteria, and algae, like I showed you, stromatolites, ruled the Earth. Then suddenly life got very complicated. That gives you an example. All these different kinds of creatures just suddenly show up. Some of these are corals, like right down here. Let's see. I think this one is called hallucigenia. It looks like it's right side up when it's actually upside down, because it has arms and legs coming out both sides. Time magazine again. Of course, understanding what made the Cambrian explosion possible doesn't address the larger question of what made it happen so fast. Here scientists slide across data-thin ice, suggesting scenarios that are based on intuition rather than solid evidence. These are sometimes referred to as just so stories. Just so because it comes completely from your imagination. This is how I think this could have happened. You don't have any fossil evidence to demonstrate that's what actually happened. This is how I think it could have happened. So, intuition, but no solid evidence. Now, Sean B. Carroll, an evolutionist, 2006, 10 years after this article in Time magazine, the explosion of animal diversity in the Cambrian is one of the most important and compelling mysteries in the history of life. Most important, most compelling mysteries. So they still don't have a clue how this could have happened, the way the evidence shows it. Then here's a quote from Paul Chen, who's an intelligent design individual. He wrote a book, Evolution Intelligent Design. And he's talking about another book here. In that book, The Cambrian Explosion, authors Douglas Irwin and James Valentine remain committed to searching out a purely material evolutionary account of the Cambrian explosion. And yet, they insist that this singular event in the history of life remains marked by important, unresolved questions, and they call the transition from sponges to the creature of the Cambrian the most enigmatic of any evolutionary transition. What's enigmatic mean?
No idea.
Huh?
No idea.
No idea?
Mysterious?
Mysterious, yeah. An enigma is something you can't understand. So enigmatic means you just can't understand it. So they're committed to some sort of material explanation, but they know they don't have one. They just don't have one. So how do you get new body plans? We talked about the difference between the arthropod body plan, exoskeleton on the outside, jointed appendages, as opposed to chordates with internal skeletons and not quite, we have jointed appendages, but not like the arthropods do. Well, you need a whole different arrangement of parts. And the developmental genes that do affect body plans tend to find their expression quite early in development. So the body plan gets laid out very early in embryological development. That's very important. And this comes from Wallace Arthur. This is a quote from him. Those genes that control early developmental processes are involved in the establishment of the basic body plan. Mutations in these genes will usually be extremely disadvantageous, and it's conceivable that they are always so. Well, that's what we have found out in the intervening years. They're always disadvantageous. So how can you get from one body plan to another just using mutations? Right? There's no indication you can do that. None.
Okay, so if there is a mutation in the first couple stages of embryological development, what happens?
Well, you either get a deformed organism or you get a dead one. In other words, it may be able to make it through the rest of embryonic development, but it won't be able to feed itself, won't be able to reproduce, and it'll die anyway. Or it dies during embryological development. Now, this is a very interesting table or graph here. Some of these, what you have across the top are animal phyla that once either exist now or once existed. The ones with a white background, those we can easily describe today. Some of these others are difficult to find in the fossil record. Tardigrada, by the way, are just really, really tiny, almost microscopic organisms, and we don't find that in the fossil record. But where you see blue, we actually have fossil evidence for this phylum in a fossil record. And also when you get down to red, we have actual fossil evidence that this existed. Now here's 542 million years ago, and here's the Cambrian, and this is what's called the Burgess Shale, that's in Canada. And you notice that there are lots of X's, things that show up in the fossil record that go extinct. They're clearly different phyla, but they all go extinct. Now, these blue dashed lines are the scenarios guided by intuition, not actual evidence. So, for instance, here we are, chordates. You come down here and there's something nearby that went extinct, and you come down here and it connects to something here, and then comes down and connects to something here. Well, we don't know what it is. Where chordates came from, we have absolutely no idea. There are no other similar forms. Cnidaria are things like jellyfish and sea stars, that sort of thing. Not sea stars, but again, they come in. You find evidence of these in the Precambrian, but we don't even know how they attach to anything else. And so you see this big category here where this leads to two extinctions and chordates. And then here from that same group you have what comes echinoderms, that's starfish. And here you have hemichordata. We won't talk about those. Nematodes, by the way, are small roundworms. And well, you see this big connection over here, and then you have all of this, supposedly all of this from here to here, coming from this. We have no idea what it is. And all you see are dashed lines. Dashed lines and then points, lines that aren't really connected to anything else. They're connected with imagination, not by evidence. And so, as I said, it usually should be that phyla come later in evolutionary development, but all the phyla show up at the very beginning. That's upside down. rather than a tree forming from a single stem, you get what they call a grass field. You have all these shoots just show up, not connected to each other. Make sense? Okay. Now, this film is going to give you a computer animated visual of what the early Cambrian seas look like.
The Burgess Shale was once part of a massive reef in the Pacific Ocean, a haven for a menagerie of life that thrived at the edge of what is now the North American continent. Throughout long periods of geologic upheaval, tectonic forces elevated these rocks and the fossils they bear more than 7,000 feet above sea level. Here, the basic body plans of major animal groups that still exist today, and many others now extinct, made their first appearance in the fossil record so suddenly that biologist Richard Dawkins noted, "It is as though they were just planted there without any evolutionary history." These fossils gave science its first detailed look at the biology of the Cambrian seas. With computer animation, we can now bring that world to life. Like something out of science fiction, Opabenia was a creature so bizarre it still eludes classification. While its 5 eyes watched for predators, the animal captured its prey with a grasping claw. First described in 1899 from a fossil found on Mount Stephen, Wiwaxia has also puzzled scientists. This mysterious Cambrian animal was covered with overlapping scales and may have fed by scraping microscopic particles off the seafloor. The animal most frequently discovered at Walcott's quarry was Morella. More than 15,000 fossil specimens of this prehistoric crab have been excavated, most revealing multiple pairs of jointed legs and feather-like gill branches used for swimming. The anatomy of hallucigenia has baffled paleontologists since Walcott first discovered its fossilized remains. 2 rows of sharp spines on its back and more than a dozen needle-thin legs gave the animal the appearance of being upside down, even when it was right side up. At the top of the food chain was Anomalocaris, the undisputed terror of the Cambrian seas. Measuring up to 3 feet long, this super predator used barbed feeding arms to capture both hard and soft-bodied animals. It then devoured its prey with layers of razor-sharp teeth.
Now, Stephen Jay Gould, again, I mentioned him earlier, and he wrote a lot of articles for Natural History Museum, and some of those were collected and published in the books. One of them was called Panda's Thumb, and he wrote these natural history articles. Okay, and what he's talking about here is he's describing how he thinks all these organisms got put together. So he tells you to imagine a large bag with 20 different compartments for 20 different kinds of structures, skeletal structure, heart and circulation, outer coating, skin, that sort of thing. And then within each compartment in the bag are about 20 different tokens of different types of circulation system, different types of outer protection. And he says that To make a new Burgess shale in Canada creature, the great token stringer takes one token at random from each compartment and strings them together. Voila, he says, the creature works, and you have nearly as many successful experiments as a musical scale can build catchy tunes. This is one of those just so stories here. He's making something up. Now, if you just take random parts from these 20 different compartments and try to string them together, they probably won't work. We're not going to fit. So yeah, you have successful experiments. Maybe you might get 1 out of 100, 1 out of 1,000 that actually would work. But he also kind of gives away the story here at the very end. Does a musical scale build catchy tunes?
No, the people who
People
use
do.
them.
Minds use the scale to make catchy tunes. They don't happen randomly. It's kind of like thinking if you put your radio between stations and all you hear is nasty static, okay, coming out of the phone or the radio or whatever, and expecting at some point in time, billions of years down the road, you're going to hear Beethoven's Fifth Symphony just suddenly rise up out of the static. It's never going to happen. Could you calculate a probability? Sure. But it's not going to happen. So we've talked about this, biological design. Richard Dawkins, biology study of complicated things that give the appearance of having been designed for a purpose. Now what does he tell you here? What does he recognize?
that it appears to be.
It appears to be designed, okay? And then I love Francis Crick, biologists must constantly keep in mind that what they see was not designed, but rather evolved. Not designed, not designed. It sure looks, no, it's not designed. Constant tension. And we've talked about this before, but a little repetition is helpful. Irreducible complexity. Without looking at your outline, can anybody give me a definition of irreducible complexity?
I guess irreducible complexity is when if you take away one component, the entire overall thing doesn't function.
Okay? The way he put it in that second section there, something is irreducibly complex if it's composed of two or more necessary parts. Remove one part and functions not just impaired but destroyed. His favorite example was a mousetrap. I think I showed you my mousetrap, right, previously. And his point simply is a mousetrap cannot be built by natural selection. Why? Because you have to have all the parts together that have to be coordinated size-wise with each other, and you can't have too powerful a spring or too weak of a spring in order for it to actually catch mice. So it takes a mind to do all of that. And the bacterial flagellum is irreducibly complex and could not have evolved without plan or purpose. Now, there's over 40 different proteins involved. If one of them simply doesn't get made, then the flagellum simply won't form. It won't complete formation. Now we come to the genetic code. We talk about DNA, the gene is transcribed into messenger RNA. Messenger RNA is translated into protein. And dozens of transcription factors regulate transcription. The three-dimensional arrangement of chromosomes is tissue specific because each tissue needs to use different genes. And so the chromosomes are arranged in a way to make sure the genes that are needed are available to be transcribed into RNA. There's other things like promoters, enhancers, repressors. And there is no junk in our genome. We are said to be 99% identical to chimp DNA, which is wrong. That's not true. And secondly, 95% of our genome was thought to be just junk. It doesn't code for genes. It doesn't code for proteins. Well, actually it does have a function. And if you take some of that stuff out, certain proteins just don't get made. What we have is a lot of regulatory information in the rest of the genome. Now I'm going to see a little video here.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Now, in the book, about the middle, he says, it eventually became clear to me that intelligent design stood as the only known cause of specified information-rich systems. What do you think that means? Specified information-rich systems. Any thoughts? Well, the word specified tells you what? It can only be one way. It's very specific. Information rich.
There's a lot of.
There's words on a page. That's information rich, and that's specified. He said those kind of systems, therefore, that ID provides the best, most causally adequate explanation for the origin of the information necessary to produce the first life. Intelligent design is the only way you get specified information-rich systems. They don't come from anything else. There are no examples of that happening. When the human genome was finished in 2001, one evolutionary scientist, Gene Myers, put it this way. He said, "We're deliciously complex at the molecular level. We don't understand ourselves yet, which is cool. There's still a metaphysical, magical element. The system is extremely complex, and here it comes. It's like it was designed." Well, maybe it actually was. Okay? There's a huge intelligence there. I don't see that as being unscientific. Others may, but not me. Now, he's not actually jumping on the intelligent design bandwagon here, because it's like it was designed. It looks that way. Just like Dawkins said about living things. They look designed. So, ID in biology. The complexity of the cell and the genome. Many regulation factors in the genome. So many different protein complexes in each cell. There is no such thing as junk DNA. We find out that the genome is hierarchically regulated and layered with information processing systems. All organisms benefit from numerous irreducibly complex adaptations that are fully coordinated. It's difficult enough to come up with irreducibly complex adaptations, but to have them all be fully coordinated is another jump. Butterfly metamorphosis. When a caterpillar builds the chrysalis around itself, by the way, all a caterpillar is an eating machine. That's all it does. Grows fat, has no reproductive organs, it cannot reproduce. When it forms the chrysalis, essentially the caterpillar comes completely apart. Almost all the cells just completely dissolve, and you have this soup of proteins and lipids and all kinds of stuff. But a few cells survive. They're called imaginal cells, and they start taking all this disjointed soup and creating a new body plan, the butterfly. Now, this comes from another Harvard geneticist, not paleontologist. This is from Richard Lewontin, and he wrote a review of one of Carl Sagan's last books called The Demon Haunted World, and he just talks about how ridiculous religion is and all these different forces out there, whatever. So I'm going to read this slowly. He says, we take the side of science. He's talking about himself and his fellow scientists. In spite of the patent absurdity of some of its constructs. There's a lot in science, he says, that just doesn't make sense. In spite of its failure to fulfill many of its extravagant promises of health and life. That's a lot of what science offered in the 40s and 50s. It hasn't happened. Again, in spite of the tolerance of the scientific community for unsubstantiated just so stories. Now, I told you what a just so story is earlier. Because, why? We have a prior commitment, a commitment to materialism. Now, he defines this further. It is not that the methods and institutions of science somehow compel us to accept a material explanation of the phenomenal world. But, on the contrary, that we are forced by our a priori adherence to material causes to create an apparatus of investigation and a set of concepts that produce material explanations. So you read science just so it would give us material explanations. He says, no matter how counterintuitive, no matter how mystifying to the uninitiated, moreover, that materialism is absolute. For we cannot allow a divine foot in the door. That's a really amazing statement. And I have a sense that once it was published in New York Review of Books, and many intelligent design theorists are jumping on that, showing that it's just materialism. And it's materialists who put that together. Intelligent design has been ruled out, not by science itself, but by the rules put in place by science. He probably regretted being quite so clear about this. Data's coming our way. Stay tuned. God is loving, caring, omnipotent, eternal, and intelligent, and God is also imaginative. Just when you think you have God all figured out, he makes a woodpecker. And you're kind of scratching your head. Why? You must have had fun doing this, God. I mean, these things are really weird.